Hi guys, so last time we started to go over our lipids and we left off with fatty acids. So I wanted to continue going over those today. Now I talked to you guys about two different types of fatty acids, but we didn't go into what was the difference between them. So there's saturated and unsaturated fatty acids, okay? And again, these are going to be our monomer of lipids. So for saturated fatty acids, we are only going to have single bonds between our carbons. Okay, so we're just worried about in between our carbons. And again, only single bonds. Unsaturated fatty acids, they are going to have at least one double bond between our carbons. It's kind of like an all or nothing thing. If there is one double bond between our carbons, it's automatically unsaturated. Okay, saturated has to have every single one of them be a single bond. So what we mean by saturated is it's going to have the maximum number of hydrogens. And if there is a double bond, well, that means it has to get rid of some hydrogens because we can only have carbon bond to four different, or we could say make four different bonds at one time. Okay, if a double bond gets in there, that means the carbon is bonded to itself twice or bonded to the other carbon twice, and that leaves less room for hydrogens to be bonded. So here is a picture of butter, guys. Okay, butter is a saturated fatty acid. All right, now we have our glycerol over here. Okay, and we have our three fatty acids bonded to it, and those fatty acids are all going to be saturated. Now, if you look at your diagram here, it looks a little weird because we just have a squiggly line here, okay? Each point of that squiggly line is a carbon. Now, with those carbons, they are all single bonded, and how we know is it's only one line in between them. If it was a double bond, then we would have like another little line, okay? Something like that there, and that represents a double bond. And we don't have to worry about triple bonds for these, okay? Because that would like end the whole chain. So, but anyway, so those are saturated fatty acids, okay? Here is a picture, again, of a saturated fatty acid. And what I did here, guys, is I got rid of the line for you. So this one had the line there, up and down. This one, there's no line. All right, a little bit easier to see that, hey, look, there's my three fatty acids on this side. There are no double bonds in between our carbons. On the other side is going to be the unsaturated fats. This one, you can see there's a kink in it, and that's the double bond right there, okay? But look here. We have three fatty acids attached to our glycerol, and only one fatty acid is unsaturated. Well, it doesn't matter. The whole thing becomes unsaturated just because that one fatty acid, all right? So it's not like a hybrid between them. It's an all or nothing thing, like I said. Okay. Now, the unsaturated fats are going to be, again, unsaturated fats. These are going to be liquids at room temperature. Okay. The saturated fats are going to be solids at room temperature. Here's another picture. Again, I just got rid of the kinks for you guys. All right. So this one here is going to be, again, an unsaturated fatty acid. But look here. Do you see the H's? If the H's are on the same side as they are in this picture, okay, we are going to call this a cis unsaturated fatty acid. All right, so cis is just CIS. Okay, that's it. It's a cis saturated cis unsaturated fatty acid. Okay. If we were to do the opposite way, so if we got rid of this guy here, which isn't working out too well for me. Yeah, it's not going to work. Okay. Let me see if I can increase this guy a little bit here. Yeah. Just pretend he's not there, guys. All right, good enough. If we were to make that hydrogen switch and be down here. That looks like an N. Sorry guys, my writing skills are off today. All right. There we go. Okay. We have an H on one side, and then the other H on the other side of the double bonds on the other side. We call this a trans unsaturated fatty acid. Just a different orientation of them, okay? Now, these are both a little bit different than, don't think of this trans unsaturated fatty acid as like the ones you hear about in stores, like trans fats and stuff like that. So those are man-made fats where they're literally like, you know, make them and market them like that. And they're very bad for you. Trans saturated fats are really bad, but don't get them confused with trans unsaturated fatty acids. They're a little bit different. Okay. So this is the one we just started talking about, guys. Saturated fats are solids at room temperature. Unsaturated fats are liquids. The saturated fatty acids are going to come from animals. The unsaturated fatty acids are going to come like from plants. That's like vegetable oil, corn oil, canola oil. Okay, they all come from plants. Olive oil, those types of things. All right. Okay, so we filled out the form last time, guys, about our lipids, and we talked about the main function of lipids is energy storage. The second one there, fat is a compact way for animals to carry their energy stores with them. What we mean by compact is there's a whole lot of bonds there, and those bonds are able to store a lot of energy. That's all we mean by compact. All right, our next polymer of lipids is the phospholipid. So what we're going to have here is, instead of like the triglycerides where we had 3 fatty acids attached to a glycerol, for this one we're going to have 2 fatty acids, and it's going to be attached to a phosphate group. Okay, and I'll show you the picture of it on the next one. Okay, so a little bit more to it. We're still going to have the glycerol in there. It's just instead of 3 fatty acids, we're going to have 2, and those fatty acids aren't going to be directly connected to our glycerol. They're going to be connected to the phosphate first. Okay. So here is a good picture, guys, of our phospholipid, and I'm going to talk about the ones that are in the cell membrane. So I'm only going to worry about our phosphate, okay, and our fatty acid tails here. So up here, the circular is a phosphate group, and it is, as it says here, it is hydrophilic. Okay. Down here, these are going to be our fatty acid tails. There's 2 of them. Those are going to be hydrophobic. So we have the phosphate that is hydrophilic, meaning it's okay with water, it's cool with water, okay? And the fatty acid tails that are hydrophobic, they fear water, okay? The other thing I want to mention about our phosphate group, okay, the fatty acids are sometimes just called tails, and the phosphates are just sometimes called the head of a phospholipid, okay? And again, guys, they're going to make up our cell membrane. So phospholipids, its main thing is to make up the cell membrane. When they're added to water, just like in our bodies, they are going to form this bilayer. So the tails are going to be in the middle, and the phosphates are going to go on the exterior wherever they're going to be able to touch water, okay? So if you look here at the layer, picture D there, the phospholipid bilayer, we have our phosphate heads on each side touching water, and then our fatty acid tails, they go toward the middle. Okay, that's why they don't want to be near water. So that's why they assemble that way naturally. All right, and here's a better picture of that for you guys. Again, you can see the fatty acid tails in the middle there. So our last thing on lipids, we're going to talk about steroids. And I'm not going to dwell on this, guys, because we don't really have to spend a whole lot of time on this. But one I want to mention is cholesterol. I know there's testosterone, there's estrogen, okay, things you guys think about. Different hormones in your body, but cholesterol is an important steroid because what it does is it helps our cell membranes maintain their fluidity, because the cell membrane is constantly moving, all right? And we're going to talk about in our cell membrane chapter later this year, what happens if we have an increase or a decrease in cholesterol, okay? Now, there's two different types of cholesterol. Too high levels of it, yeah, you could have heart problems, cardiovascular disease, not good. All right, our next one, proteins. Let me go back to our chart here. We'll draw some things out for proteins here. Okay. Our function of proteins, there's going to be a lot. We're just going to say building blocks right now. Okay, because there's so many, I'm not going to be able to fit them all in there, guys. Our polymer our monomer of a protein is called an amino acid. And then once again, we are going to have two different polymers. They could be a dipeptide or a polypeptide. All right. Okay. Structure of a protein. Let me enlarge this a little bit. Okay. In the middle of our protein, we're going to do a few different parts to it. In the middle of our protein, we are going to have, find where my box went here. There we go. In the middle of our protein, we are going to have a central carbon. So draw my carbon in there. Easy enough. Okay. On our central or alpha carbon, we are going to have a hydrogen attached. Cool. On one side, we're going to have a functional group, an amino group. Okay, that's our amino group. On the other side of it, we are going to have a carboxyl group. Okay. Now, before I put the last thing on here, guys, let's talk about this for a minute. We have our carboxyl group on the one side, our amino group on the other. Let's talk about the functional groups. If you can remember our functional groups we went over, I think on the first day of this chapter, the carboxyl group makes it acidic. The amino group makes it basic. Okay? Acidic on one side, basic on the other. Pretty much the amino acids, guys, even though it says acid in the name, they are going to be very close to being pH balanced, okay? So close to 7. And then up top here is the R group, all right? The R group is going to be our variable group. Right? I don't want to draw a line coming out of the R so you guys don't think something's connected to it, but this is our variable group. And there are 20 different R groups for the amino acids, guys. So there is your structure of a protein, your structure of an amino acid. All right. Let's get rid of this. Okay. So functions, guys, of proteins. Yeah, there's a bunch of them. Okay. Here's a few. Proteins act as enzymes. They could be your antibodies to bad bacteria and bad viruses. Okay, they help to store things. They help to transport things. They're embedded in your cell membrane. So like I said, guys, all the functions weren't going to fit in that little block we had there. They help to transport things from one side of the cell membrane to the other side. They help with hormones, okay, and movement of hormones. They could be receptors in your body. Maybe like, if you're having some pain, okay, you hit your arm on something. Those receptors will be like, hey, we're feeling pain. Take it easier there for a while. They're going to help certain things move, maybe in your bodies, maybe in bacterial cells, like the cilia, the flagella. Cilia is even like lines your esophagus going down to your stomach, and your trachea going down into your lungs. But yeah. And then there are things like collagen, like in your skin, guys. So there's structural proteins there. So let's talk about enzymes for a little bit. Enzymes what these do, guys, is they are going to lower the amount of energy we need in order for a reaction to occur. Okay? So the enzymes are going to help us with keeping our body going without using as much energy. In living things, we call them enzymes. In non-living things, they're catalysts. A catalyst and enzymes are the same thing, just one's in living things, one's in, not living things. But the nice thing about enzymes and catalysts are we can keep using them over and over and over again without them being consumed. In other words, they're not going to go bad unless they do, and then we make a new one. So we talked about the polypeptides a little bit, guys. There are 20 different amino acids, okay? So 20 different R groups, which those are the 20 different varieties we have to choose from in order to make our polypeptide, okay? We talked about this one, guys, the R groups. So there is your structure of an amino acid. There's the alpha carbon in the middle. There's everything that we drew in our diagram. Here are all of your amino acids, and your book separates them very nicely, actually. So everything in yellow, those are just the R groups, everything in yellow is going to be hydrophobic, okay? Now, I'm going to let you guys look over these later on because there's a question on your assignment with them. But if you look at these, you'll be able to sense a pattern with why those R groups are hydrophobic and why these ones are hydrophilic. For the most part, you can. Okay. And there's actually one in here that I was looking at last week, and your book is like not wrong with it, but sometimes one of these is going to act a little bit different, or it may not be completely hydrophilic. These ones here, guys, they could be acidic or basic, all right? So you could look over those as well. So there's all your amino acids. When we are going to put our amino acids together, when we're going through the condensation, dehydration, synthesis reactions, we are going to make a peptide bond between them, okay? These peptide bonds are going to connect a carboxyl group, they call that the C-terminus, to the amino group of the next amino acid, okay? So it's never going to be carboxyl to carboxyl or amino to amino. It's always going to be one to the opposite. Here's an example, guys. All right. And do you have to know all 20 amino acids? No, you don't have to be able to recognize them. In college, you will, if you're going for like a science major, but for now, you don't need to worry about them. Okay, protein folding. We'll go over protein folding and then we will call it a day, guys. So we're going to go over how a protein becomes folded from when the amino acids start to bond together. Right? There's a few different like levels of protein folding, and it goes to 4 different levels in order to make its 3-dimensional structure. So here is the antibody of a protein. And you guys can see that there is this area right here, and it attaches perfectly to this protein from, a flu virus. So what that does is it allows us to take the flu virus in our bodies to get rid of it, as long as we can know it's foreign, so if we have those antibodies. But anyway, 4 levels of protein structure, guys. We have primary, secondary, tertiary, and the last one is quaternary, which is separated into a different one on here, and we'll talk about why that is. But here's a short video on the protein folding.
The shape of proteins. There are 4 different levels of protein structure or protein shape: primary, secondary, tertiary, or the 3rd level, and quaternary, or the 4th level. The primary or 1st level of protein structure is a chain-like or linear sequence of amino acids joined by peptide bonds, as you can see here. The secondary or 2nd level has 2 shapes: an alpha helix or coil-like formation, or a folded pattern called a beta sheet. In both of these structures, hydrogen bonds between the R groups of the amino acids stabilize the shape of the protein. The tertiary or 3rd level of protein structure involves more folding and bonding of the secondary structure. The coils may even touch each other, as some covalent bonds form, from the sharing of electrons between different amino acids, but most of these twists and folds occur from the result of ionic bonds between positively and negatively charged R groups of the amino acids. The quaternary or fourth level is where clusters of more than one polypeptide chain link together to form a giant molecule. Many different types of bonds may be formed within this structure. The shape of protein is important. For instance, fibrous proteins are extended linear proteins that are part of ligaments, tendons, and muscles. And globular proteins fold into almost spherical shapes and have their hydrophobic or water-fearing R groups buried deep within the core, and their hydrophilic or water-loving R groups extended out into the water when in an aqueous environment. Proteins take their shape based on the job they're required to perform, and some have moving parts that are important to their functions. Proteins can have their structure or shape altered by denaturation. When this happens, the protein is no longer able to carry out its job. Denaturation can take place because of change of temperature, change in pH, or the presence of certain hazardous chemicals. And that be the basics on the shape of proteins.
All right, so short video, guys, like I said. So here is our different levels of protein structure. So primary, pretty much, we're getting these amino acids, we're getting these monomers, boom, we're binding them together, that's it. Nothing too crazy there, okay? Secondary, we are going to start to coil and fold our polypeptide chain. So not much interaction has happened yet between the adjacent R groups, except for the actual amino acids being bonded together from the C terminus to the N terminus, and that's it. So that's all secondary is. Tertiary, this is the first area where we start to have an actual three-dimensional model of our protein. So the R groups, having their different properties of the R groups, whether they're hydrophilic or hydrophobic, this is going to determine how the protein is folded. If we have adjacent R groups being both hydrophilic, they're going to get closer to one another, okay? If they are hydrophobic, they're going to get closer to one another. But if one's hydrophilic and one's hydrophobic, they're actually going to try to repel one another. And that's going to be this whole gigantic long chain. The whole chain is going to start to fold based on these hydrophobic, hydrophilic interactions. And that's going to make a three-dimensional model. Now the quaternary one, the last one, is going to be when we have more than one polypeptide chain. They're all going to come together and form an even bigger protein, okay? So the quaternary one isn't too much of folding. The tertiary one is where we actually get our three-dimensional structure. The quaternary one is bringing like more polypeptides in and making, you know, a bigger model. So here's our primary, guys, all right? You can see all of our amino acids are bonded together. We get our secondary where it starts to coil. Our tertiary, okay, which again, we have our three-dimensional shape in tertiary. First time we have this, we have the R groups interacting between one another. And then the quaternary is going to be, you could see, more than one polypeptide there. All right, last slide of the day, guys, and then we'll talk about a little bit more with proteins, with enzymes, and we'll go over sickle cell tomorrow. But collagen, you guys can see there are three different proteins in here. It forms like a triple helix kind of thing. So collagen has really, really, really good strength, and it's a really good protein. It makes up things like your skin, your bones, your tendons that connect muscle to bone, and your ligaments that connect bone to bone. So obviously we want all that stuff to be very, very, very strong, right? Especially your skin. You don't want your skin to like just start sloughing off. That would be kind of gross, right? But anyway, so collagen, extremely strong protein, and it's in a triple helix structure. All right, guys, have a good rest of your day.